Okay.
I think this is the right moment to do it. So
yeah, well, it's pressed now, so thank you. So, Bets, you are now live, as it were.
We're now live. We're going to be recorded and then be dead or something. But so thank you all for joining us. And so our first speaker is going to be Courtney Davis from Janssen Pharmaceuticals, who's going to speak for 25 minutes, and then Natalie Dean from the University of Florida. And then we have a few discussants, and then we'll open it up for open discussion. So Courtney, are you going to share your screen or do you have slides?
I am. I'm going to share my screen so you can see my slides, which hopefully will work.
I see something. Yes, something is showing up. Yes, wonderful.
Yes. Promising.
Promising. Yes.
Fantastic. Fantastic. Well, thank you so much, Bets, and to this very distinguished group. It's quite a pleasure to join you today and share with you some thoughts about real-world evidence and decentralized clinical trials in the context of COVID-19 vaccine development. And full disclosure, much of this content was presented in the fall at an FDA workshop, but I have updated it to try to make it reflect, you know, the current status, because this is such a rapidly evolving area. I am an epidemiologist by training, and I am a member of the epidemiology department at Janssen R&D. And there's my disclosure. So I'm going to cover in 25 minutes or less 4 main areas. Briefly talk about the limitations of conventional randomized clinical trial designs for a COVID-19 preventive vaccine, and then talk a little bit about decentralized clinical trials and why those are appealing in this context. Specific opportunities for how to incorporate decentralized clinical trial and real-world evidence components into a COVID-19 vaccine study design, and then end talking about the current challenges and future directions of these concepts for this specific vaccine kind of design, but also just thinking about any kind of pandemic vaccine development program. It probably doesn't need to be repeated, but I will just start with the context for this pandemic. When we started working on our vaccine development program last spring and summer, it was very acutely obvious that sort of conventional clinical trial designs, which require frequent clinic visits, you know, are usually highly selected patient populations, and rely upon a concentration of sites which are based in academic centers with, you know, excellent clinical trial capabilities, weren't going to work so well because we had to face social distancing and travel restrictions, where we had to reduce unnecessary contacts and to minimize the risks. We also needed to have flexibility because we knew there would be variability in policy and behavior changes that would be required. We had this incredible urgency to try to respond and also figure out how to have very large trials that could be scaled up in a really short time frame. And then we had to address populations who are at higher risk, who historically are much harder to get into studies, including the elderly and minority groups who also don't trust clinical research as much. We also wanted to try to minimize disruption to people's lives, you know, as a way to make participating in clinical research more appealing. And just highlighting here some results of a study that was done by J&J to try to understand why volunteers would or would not be interested in participating in clinical studies. And some of the factors that were most important to people who are considering participating in clinical research were whether they were going to have to travel, whether they would or would not be able to do any kind of testing from home, what the overall time commitment would be, and whether they would be able to have, you know, fewer requirements to meet in person with research staff and would be able to make use of clinical innovations that would be digital in nature. So the context of the pandemic needing to have huge trials, fast, flexible, include populations who weren't usually in clinical research, and also the need to appeal to volunteers in very large numbers brought to mind these decentralized clinical trial concepts. And just to, again, level set in terms of what a decentralized clinical trial is, they're defined as those that are executed through telemedicine and mobile local healthcare providers using procedures that vary from traditional clinical trials. And they're also sometimes referred to as direct-to-patient, patient-centered, remote decentralized trials or digital trials. So just trying to get the terminology straight out there. We were thinking about these DCTs because first, the regulatory framework has evolved where these fit nicely in the real-world evidence framework that's been outlined by health authorities, including the FDA. From a technology standpoint, the technology is continually evolving and improving so that we have the ability to do decentralized clinical trials with data integration and also more tools that have been validated for use in, in something like a phase 3 pivotal study, including tools for electronic consent, for digital engagement of patients, and for data collection of various different types direct from patients or from sensors or other devices. And then finally, this really fits in this patient-centered focus type of framework, which is part of the 21st century cures. Here's another, graphic where we're trying to highlight where these different elements fit in the context of COVID-19. And if we start on the left side of this diagram, these DCTs can be ways to help us improve digital outreach and screening. Because we were trying to reach large numbers of people, but also special populations, we could make use of existing digital communities, for instance, and also use healthcare data to screen for patients who would be at high risk and would qualify for the trial. And that should speed things up, right? Both speed things up, but also get us a higher risk population, which allows us to not just find the people, but then have a shorter trial, because if we have higher risk people in the study, it should go faster. We could also have participant input into the design. So if we can reach out and connect with these communities, we could get feedback on the design of the study, which should make it more appealing and, again, more relevant, and also go quicker, as well as being convenient and appealing to the patients. So their participation is enhanced. Moving all the way across, I think I've already covered, it's the ability to make use of the technology, which again drives down costs, but also makes it easier for patients. And then finally, in the end, if we've been able to recruit a high risk population that's relevant, we're also hopefully helping to address health disparities by having included a more diverse participant pool into the trial. So some of the challenges we face, I've got on the left column with what we're calling the opportunities on the right to try to solve the problems. So we are worried about inefficiency due to recruiting a large number of low-risk volunteers. We know we can use digital solutions to get a lot of people. There's lots of success stories of using social media to recruit patients, and Apple Watch to conduct a trial. But we needed to find high-risk people. And one way we could do that was to analyze real-world data that already exists, healthcare claims, electronic medical records, cohorts that were already in place, and then screen using that existing data, and then do the targeted outreach. Another challenge we faced was this lagging recruitment of diverse populations. and including, their hesitancy to participate for very valid reasons. We actually chose to address that through partnering with investigators who themselves are diverse and or are located in diverse communities and where they already have deep existing relationships and trust, and working through those community leaders. Another challenge is loss to follow-up in long-term trials, and these trials need to follow up participants for up to 2 years. So the opportunity to solve that is thinking about nesting trials within existing healthcare delivery systems. Again, in vaccine trials, maybe this is not so unique because there's a history of doing this in some instances. But maybe in the U.S., especially in a phase 3 situation, this is not so common. We thought about that from the start, at least in the initial part of identifying potential partners who had access to the data, healthcare data, to screen and recruit. If we had that continuous data sort of backbone throughout the trial, you could see how it could offer flexibility to actually substitute some of that routine data that's collected and avoid follow-up visits, because you'd be picking up any of the, for instance, healthcare encounters related to COVID-19 or related to any severe safety events. Those should be picked up within the regular healthcare system and linkable. Consenting to link to the real-world data up front when you're designing the study, again, offers a huge opportunity to simplify the data collection. Again, less burden for the investigator, less burden for the patient, and actually potentially more complete recording when it's all coming directly through the healthcare system. And finally, we could, across the different studies, meaning across the different vaccine manufacturers, there's also an opportunity to harmonize the endpoint definitions using the same codes or algorithms using the real-world data. Another challenge we faced, all the manufacturers faced, was making sure that there would be specific codes for our vaccines that would be available immediately post-authorization. And the reason this is a concern is that if we want to rely upon the real-world data that would come after the vaccination, as we're continuing to monitor the safety and effectiveness, we really need to be able to be specific when we link the outcomes to the vaccine, especially knowing novel platforms are there, and we want to be able to differentiate them and not just pick up a code for any COVID-19 vaccine. The good news is that we did get out in front, all the manufacturers, in making very clear this need, that we've been reassured that those specific codes are available. In fact, we were hoping that they would be mandated to be used in the claims for reimbursement. Again, as another way to confirm they would be present, is the best way to do that, is requirement for the doctors, health systems to get paid. They have to put the code. And then finally, another challenge would be capturing events outside of the usual healthcare encounters. And I mean, I think this is still something we're very much monitoring and I would say not totally sure about, which is when vaccine events occur not in a usual healthcare environment, meaning large outside, large tents or mobile vans, or the employer-based vaccination programs. We understand that the cards are being given to each participant who's vaccinated, but how will that make it into their healthcare record is something we've been banging the drum about. It's early in the emergency use period, specifically, that is the concern. Later, when these are under the full licensure, they're probably going to be in normal healthcare settings, and that is more typical where we would see the claim come through. In general, the opportunity would be, ensuring there's a mechanism to link those exposure details into the patient record. And there are digital solutions to that, but it's not going to be as helpful if those aren't uniformly done across all the different manufacturers and settings. So this is not a professionally done graphic. I take full responsibility. An epidemiologist trying to do PowerPoint is sometimes a scary thing, but I did want to try to give you a visual of what the study design looks like, kind of currently, and then where it's moving towards. So if you look on the far left, we're starting with populations. We're using data to screen them, and we're sort of nesting, when possible, we're nesting our recruitment efforts in data systems. And as we do that screening, we can efficiently find the right people, get them in to be consented and randomized. And then we can monitor them over time with a combination of visits to the facility, but minimize those. So those little circles with the building are at the initiation of the study, and also at the end of the study. But ideally, in between, we could use a system of telehealth and nearby units where folks could go to have the confirmation of infection, for instance, if they have symptoms, to have routine blood draws, to have any kind of swabs, et cetera, or home kits could be used. That would allow us to follow up efficiently on any important safety events or important efficacy endpoints, but try to minimize the churn for the patient. And then over time, as EUAs and BLAs occur, people might, if they're in a placebo-controlled trial, might want to exit. But that's not unexpected, and we could account for that either in the trial design, of course, and that's a whole nother topic, of people crossing over within the study, but also some people could leave the study to take some other product which has become available. But then we would still, if we had access to monitor their real world data over time, we would still be able to pick up all the major events without them ever coming back. Even if they exited the trial, for instance, we might be able to continue to monitor their major events, which is a lot more efficient than having them completely lost to follow-up and no information available. The beauty of continuing to follow their health record is we would know not just what other vaccine they may have received, but then also all of their subsequent events. That's just one example. We have some elements of this in the Janssen trial, and our other partners in Operation Warp Speed, other manufacturers also have some of the same elements. Moving forward, though, I wanted to put out there what I think of as more of an ideal future vision, and that is, when you have a little more time, then you can think about not just, I would say, the quickie version of the readiness cohorts, which is what we had in our study, but more thoughtfully planned readiness cohorts, knowing where these higher risk events occur. We know that they occur in occupational settings, they occur in neighborhood settings, and therefore we could think about building readiness cohorts, working together with employers. Also working with communities, and that can be done also through healthcare systems which are embedded in communities. The advantage of getting in front of this much earlier, of course, is that we can both do surveillance to understand risks over time. And then we also could get that pre-consenting done for all the data linkage. That would put us in a much better position to understand the complete longitudinal experience of persons who are vaccinated and people who are not vaccinated, people who intentionally are delaying their vaccine, for instance, which we're already seeing happen even in healthcare providers and healthcare workers who we thought being offered the vaccine first would all be jumping at the chance. We're seeing, in fact, actually not all of them are willing to do that. If we pre-consented all those people and we're able to follow them, think about what a great position we would be in to be able to have external control groups, for instance, ready to go. So that in addition to, of course, continuing to validate safety endpoints, validating effectiveness endpoints with the same real world data sources we want to use for our trials, puts us in a much better position, again, to be efficient with using the real world data and minimizing the prospective collection and invasive collection of our volunteers who we really depend upon when we're doing these very large studies. So I just want to end with my conclusions and next steps. We've learned a lot in the last 9 months about conducting research during a pandemic. I think the good news is that in this situation, we really had to accelerate innovation, but we saw how much it was possible when everyone, including the health authorities, the community leaders, the healthcare practitioners, everyone wanted to remove all the barriers and embrace innovation. We were able to get lots of things done we never thought possible. We also think a lot of these innovations will help us increase diversity of participants, and that's an important goal, not just for COVID-19 studies, but also for all research moving forward, because that's lagged. And again, I think that the pandemic, has necessitated movement forward in that direction. I also think incorporating distributed real-world data networks and methods, including common data models, is going to enable us to be more impactful with real-world evidence, meaning we could be more efficient during trials as well as post-authorization studies. So again, using epidemiology and observational designs has always been accepted for post-marketing, especially safety studies. But I think that this pandemic has shown, really we can move that much earlier and still have robust evidence. Finally, partnering with the communities, including employers, really is something we should continue to work on to develop readiness cohorts in advance. We need to build trust with communities. We need to partner to make sure our studies are going to be meaningful and feasible. And part of what we have to keep working on is vaccine hesitancy, because it doesn't matter how fast we can do a trial. If we can't get people to take the vaccine after the trial is over, then we're not going to be successful. So we need that whole arc. We need to pull out all the stops for that. And finally, I think we still need additional guidance from our health authority partners on what data sources and endpoints are acceptable, so that we can actually, continue to design and deliver research which will be successful. That's it from me.
Well, thank you. Thank you, Courtney. I guess you can unshare your screen now. So thank you, Courtney. That was an excellent talk. And your challenges slide reminded me that I forgot to say something at the beginning, that one of the goals of this session will be ultimately to write a paper for in the sequence on the challenges of vaccines in the pandemic situation. And so you have already had a slide on some of the challenges. So, and thank you for that. So we're going to go on before we start discussion and ask Natalie Dean to be the next speaker. And Natalie Dean is an assistant professor in the Department of Biostatistics at the University of Florida. And she's going to talk on what's on her screen. So Natalie, take it away.
Yeah, hi. Yeah, thanks so much for inviting me. And yeah, thanks in particular, to Bets. Bets and I work closely together. And so she's been involved in a lot of this stuff I'm going to talk about. And so I took the liberty of talking about, not specifically about COVID-19. Beth said that was okay. So I'm actually going to talk a little bit about our work that's been ongoing for the last 5-6 years, thinking about vaccine efficacy trials for more generally different types of emerging pathogens. Okay, so I've been part of the WHO's R&D Blueprint Initiative since its inception. This is the Blueprint for Action to Prevent Epidemics. We've been part of the subgroup on developing norms and standards for clinical trial designs for evaluating new vaccines and therapeutics during the context of public health emergencies. And so part of that blueprint, they've identified a set of prioritized pathogens considered likely or have potential to cause public health emergencies and for which we lack adequate medical countermeasures. So these include some more familiar than others, but Ebola, Marburg, Crimean Congo hemorrhagic fever. It included the two severe coronaviruses, MERS and SARS, Zika, and then also disease X. So it recognizes that a future pandemic could be caused by some as yet unidentified pathogen. And so we've been thinking sort of very generally about some of these problems for a while, and we have some sort of general considerations. This is a product of our working group. You know, what are some of the endpoints, target populations, unit of randomization, statistical analysis, and really laying out some of the major challenges. So in keeping with the focus of this session to think about challenges. And I mean, the big challenges for running vaccine efficacy trials during public health emergencies, it really boils down to well, first of all, these compressed timelines for writing, approving, and implementing protocols. And our previous speaker did a great job explaining some of that and the practical issues. And then also what I'm really going to focus on today is the unpredictable spatiotemporal incidence of disease. And so again, I'm not talking specifically about COVID-19. COVID-19 is a little unique in that it's everywhere. So that's really actually made it, when we think about how quickly these vaccine efficacy trials have moved, they've moved very quickly because there's just so much transmission going on. But for the other diseases that we've been focusing on for a while, that's not always been the case. And really, when we think about assessing efficacy and running endpoint-driven trials, so these trials need to be placed in the right locations. It's really hard to predict where outbreaks will occur. I'm thinking about Nipah, I'm thinking about chikungunya, I'm thinking about these sort of boom and bust outbreaks that occur, these pop up in these areas, and it can be very hard to predict in advance where that's going to occur. And at locally, transmission may be quite high, but when we think at a larger population level, when we think about prospective decisions about where you're going to run a trial, that incidence can be very low. So unlike for endemic diseases, there may not be sites with reliable incidence. It's not like malaria. You can set up a trial and eventually there will be enough transmission. You could set up, an Ebola vaccine trial and there could, never be transmission in an area, at least not in any feasible timescale. So the trial really needs to be placed in the right location at the right time. And how do we make these work? So this is some of the strategies we've been thinking about for the last few years. I'm going to go through 3 major approaches. And the first is the concept of responsive design. So these are flexible trial designs that follow the outbreak as it evolves. So the typical strategy for vaccine trials, I mean, in the previous speaker, I mean, there's obviously practical considerations and so we're, figuring out sort of what is possible in these settings, but really, in general, you pre-specify a set of vaccine trial sites that you expect to be stable throughout the course of the conduct of the trial. In responsive designs, what we're talking about here is a setting where the locations of the trial, the populations you enroll, the locations you go to, these are not sort of fully pre-specified at the start of the trial. And so you have some flexible element that allows you to follow the epidemic as it progresses. What we're talking about really is best for fast-acting vaccines. If you have a 2 or 3 dose vaccine that takes a while to kick in, this, you really, in those situations, you'll need more lead time than this will enable. But if you have a fast-acting vaccine, and particularly if you have pathogens that sort of move through predictable networks, this can be a useful approach. And so the example I'll give, so we were involved in a ring vaccination trial design for Ebola that was run in Guinea during the West African epidemic. And this used ring vaccination, which was used to eradicate smallpox in the 1970s, and sort of converted it to a clinical trial design by adding randomization. And so the basic concept is that you have a newly identified case, and then you identify the contacts and the context of that individual, and that forms a unit. It could be a spatial unit, but here it's like a contact-based unit. And then you that unit then is randomized as to vaccination or some control. You could individually randomize, but in this setting, it was a cluster randomized trial design. And so then mobile teams went out to these locations and enrolled participants. So sort of, it's been described as a trial in a box. So it required some sort of, technology to maintain the cold chain, but I think, it really demonstrated what was sort of possible to achieve even in a setting that there's not a lot, during an active public health emergency and without a lot of experience with clinical trial, conducting clinical research. And so the advantage of this is that, so here's the epi curve for Ebola in Guinea, and the decision to conduct the trial was around the peak of the epidemic there. But by the time, it takes time to get everything set up. And by the time the trial started, incidence had started to wane. But because the design was so highly targeted, even though there weren't so many cases after that point, because the design was so targeted at where exactly these locations where transmission was ongoing, a good chunk of these cases were either index cases or secondary cases in this trial, and so contributed to the assessment of efficacy, and that vaccine is now licensed. The vaccine also worked well, so that helps too. So, so other responsive features that we're talking about, and so we've been talking about these for chikungunya and other types of viruses, thinking about creating a network of pre-specified sites. This actually relates very nicely to the previous speaker's discussion about these cohorts sort of in place. But you have this network and you increase surveillance at these sites, so you're kind of ready to go, but you only enroll participants or vaccinate following some sort of surveillance trigger. So you have a population, but you wait for some conditional, something needs to occur before you actually go in and vaccinate to preserve resources. Another approach is to include mobile trial teams to enhance flexibility. So I think there's a lot of approaches here that can make so you can be more targeted in where you place the trial. Okay, so that's sort of a general discussion of that first element. The second is thinking about how we can integrate modeling. And so I know this group has a lot of great modelers, people interested in modeling. And so I've been really thinking about, or our group has really been thinking about the intersection between modeling and clinical trial design, specifically vaccine trial design. So how can we use data-driven approaches, particularly to guide site selection, but also to guide a number of different decisions? So I'm going to give the example of Zika. So again, not talking much about COVID-19. So the motivation here was thinking about designing an individually randomized Zika vaccine efficacy trial. And the challenge here was to identify areas, so sites, potential trial sites, that had sufficiently high projected incidence that could be explored for inclusion in a vaccine trial. And so there were a number of different, modeling groups that pursued sort of this problem that was, asked of them by NIAID. And so, the goal here is you have these spatially resolved models. So these models that can identify sort of differences across locations is actually integrating real, local data about sort of which sites are at increased risk for future transmission. And I'm going to talk briefly about a paper that used 3 separate modeling teams that were tasked with ranking sites by country for several different countries in the Americas, and then they synthesized the results across those teams to make some predictions. So I'm using this example. This is a preprint, and there were 3 different spatially resolved models. The first is a meta-population model that used mobility patterns. So we have these network of sites that have some has some connectivity, some movement between them. There's a model that identified sites at risk where much of the population remains susceptible. So thinking about serology or where the sites where transmission had already occurred were kind of removed from consideration. And then there's another site that leveraged another model that leveraged similarities between patterns of spread for Zika and dengue. So places where there had been a lot of dengue, but were considered to be at risk for Zika. But if they hadn't had a lot of Zika yet, then that would be a place that would be considered at risk. So one of the models was a model that we've been working on. So Betsy and I have been a part of working with our colleagues at Northeastern. And so this is the meta-population model. And so this integrates a number of different data layers. So you can use data, spatially and temporally resolved data on mosquito abundance, temperature, as that sort of can modify the mosquito lifespan, mobility and connectivity between locations, socioeconomic status, where that modifies risk. And then you can use that to make projections about areas where you think there's the highest projected incidence. And so that was one of the models that fed into this project. And so you can see, you can start to use sort of a data-driven strategy to identify areas that are at highest projected risk and explore those for inclusion in a site. So in a vaccine trial. So here we have Colombia and Brazil. So we dug into this a little bit more. And so this is a paper that, so my wonderful postdoc, Zach, is on the line, just submitted. And so one thing you can do, so we can think about starting to, how do we prioritize sites for selection? How do we identify some of the optimal features for a study design that really uses what we know from the model, or what the model tells us about these probabilities and these relationships between sites. So you can identify different ways to sort of rank sites and identify sort of top ranked locations. And then you can explore sort of what is the relationship between this modeling and the optimal trial design. And so one thing we explored is kind of decisions about how many sites to include. And so let me orient you to this figure on the left. And so here we have, imagine you have a fixed number of total participants. You have 10,000 total participants. So you can simulate trial designs where you have one site with 10,000 participants or 10 sites with 1,000 participants each. And then you can see sort of what are, from the simulations, what are the expected number of cases that you would observe in a trial population sort of in these different locations. And what we found is that, as expected, if you have only a single site, if it's the highest risk site, then, there's some probability that you have a lot of cases, but there's also a big probability that you have no transmission. I think that's the big risk in this setting, is that you can end up placing a trial somewhere where nothing happens. And so we can explore then, as you add more sites, you're less likely to have transmission take off in both places or in all 3 places. So this comes down in terms of the immediate, this expected number of cases. but you reduce this probability that you have no expected cases. And so you can sort of frame that as an optimization problem and start to identify, maybe you need roughly 10 sites. This is kind of a simplified example, but it shows the type of thinking that we're following. You can also use the model to explore correlations. And here we have a metapopulation model that has this network of sites that may be similar in terms of they're close to each other, or they have similar geography or climate, so similar risk. And so we can look and see this correlation within a correlation across sites. We can see here, this is all, for Mexico. These are all the different sites. They're sorted by latitude. And so we can see that like within Mexico, the sites tend to be highly correlated. And so that can also have bearing on the design decisions as well, because when we think about selecting sites, it may actually be advantageous not just to look at the highest rank, but to also pick a design that treats units as kind of independent units, because if you pick two sites that are close together, even if they are both highly ranked, if they're sort of moving as dependent units, then there's an increased risk that there's no transmission there. And so the way we kind of thought about that is, here's the probability of sort of having enough transmission at your selected sites. If you pick 3 sites, these are the 3 highest ranked sites, but actually there are lots of different other combinations that perform better. And we found that often these are combinations where the sites are geographically further apart. And so here, with 6 sites, here, these are more geographically dispersed. So this is another opportunity to think about using the modeling to make these design decisions, sort of explore the space of possibilities. We recommend the use of ensemble modeling. So not just using one model, because any one model may have strengths and weaknesses. So definitely recommend having groups contribute, independent groups contribute, so that you have sort of a more robust projections. But this can be very useful, we think, at the beginning of a trial to guide decisions about site prioritization and selection, throughout the trial to make decisions or to assess the expected duration of the trial, or to make decisions about feasibility. When incidence wanes, so say you have transmission slowing down at certain sites, you may use this modeling framework then to decide whether you need to sort of change course and add new sites. And I think this could become relevant in the US, if we, for COVID-19, I mean, if we do see that transmission, fingers crossed, like if we're able to bring something, achieve better control with the use of vaccine, then there may be decisions about to move the trial in other locations and what's sort of possible. Okay, so keeping an eye on time. So the last thing I'll talk about are core protocols. And so basically the key here is, what can we do to facilitate collaboration and to combine efforts? Because we think this is an important element of a successful design. And so this comes back to the Ebola experience. And this is a figure from a report from the National Academies with this idealized timeline of how you conduct a clinical trial. And the idea is that as the epidemic starts, then you want to accelerate your processes. So you launch your trial before the epidemic peak, so you can hit that or capture that peak period of transmission to be able to tell if the vaccine protects. But one challenge here is that when you're here and you're making these planning decisions, you don't know whether you're going to have this big epidemic or something quite small. And we have seen that with Ebola, that, like in the DRC, not all of the outbreaks over the past few years have really taken off to the same extent. So, when you're sitting right here, what kind of decisions these are real practical questions, what kind of decisions should you be making about running a trial? Is it worthwhile? And so after a lot of discussion, our group really thinks that all outbreaks represent an opportunity to advance R&D efforts. But, the concern is that for small outbreaks, there's considerable risk that trials will need to be terminated early due to low accrual. So the epidemic ends there, And so you have these underpowered, inconclusive results. And prejudgment of these types of results, so I mean, the standard has been to publish sort of whatever you have at the end, but prejudgment of these promising but inconclusive results can impact decision-making of caregivers and policymakers. It can also get you into this sort of evidence limbo where then no one is willing to sort of conduct future confirmatory trials. And we've seen that with some of the monoclonals for like ZMapp for Ebola. So there is a sort of a complicated decision there. And so this kind of happened with COVID-19. I mean, remember way back when China was the epicenter of the pandemic, and but then incidents waned, but it took off everywhere else. And so back in February of last year, a lot of treatment trials, so therapeutic trials, were started. But at some point, incidents waned. There weren't people, requiring hospitalization or treatment, and so those trials stopped. And so we ended up with, very informative but underpowered results about evaluating these different interventions. So, this paper we wrote. This is one of our products of our blueprint efforts. This came out in April, but we submitted it a full year earlier. We've been thinking about this for a while. So we really recommend designing trials that can extend across outbreaks or extend across locations. One big trial being better than many smaller trials. For many reasons, it can accrue evidence faster, but the main overarching motivation is the framework is that trial results are released only following the advice of an independent data monitoring committee and not simply due to lack of recruitment. And the goal is to preserve data confidentiality and data integrity until the scientific aims have been reliably addressed. So in the basic idea is that we're thinking about one sort of large mega trial where you have sort of unified oversight to avoid the situation where we have many underpowered trials, or the situation where we have to wait for each of these independent trials to read out, which could take quite a bit longer. And in the context of a pandemic where we want timely decisions, that can be quite a bit less efficient. And so 2020 has definitely been the year of the mega trial. And I think it's really, been a lot of really incredible advances, and I think a lot of great lessons learned in how to conduct this type of research. So, so wrapping up, unpredictable spatiotemporal incidence is really a key challenge for assessing vaccines during outbreaks, unless it's COVID-19. But I think in the future that could become more relevant. The recommended strategies we've been thinking about are using these flexible, responsive designs, using modeling to inform sort of data-driven planning decisions, and collaboration under a core protocol or a master protocol framework. And yeah, just, I'm excited to chat about this with folks. I think there's a lot of room for innovation in the design of these. So, so thank you so much for listening.
Well, thank you, Natalie. That was great. That really sets us up. And I see that Jamie has joined us in the meantime. So both the talks were really innovative and forward-looking. So I'd like to open it up, first of all, to the listed discussants who are not obligated to say anything if they don't want, but they can. So the first one on our list is Jesse Berlin. From Jesse, would you like to make any comments either on the talks, or it can be also independent comments?
Sure, thanks, Beth. For those who don't know, I head epidemiology for Johnson & Johnson across pharmaceuticals, medical devices, and consumer products. But I've spent a fair amount of my time in the last year thinking about COVID-19 vaccine trials. So I'll just more or less supplement some of Courtney's comments. That for some background, I don't know how closely people have been following the discussions in the US around these emergency use approvals, but there have been 2 FDA advisory committee meetings that both took place in December, and that's where the discussion took place about should this be approved and what do we do? You know, how do we implement vaccination? What do we do about follow-up? And one of the issues that became primary in those committee meetings was what to do about placebo groups. And now look at the situation. We have 2 vaccines that are approved for emergency use, and you have ongoing trials. So the whole question of how long do you keep the placebo group going exposed only to placebo before there's, well, there are obvious ethical issues that come up, and there are scientific issues that are kind of running along with those in parallel. So I'm not going to get into those in too much detail. I will refer you to the FDA website. We'll have recordings of both of the advisory committee meetings. And one of the talks that I found most interesting was from Steve Goodman, who's at Stanford, who talked about this whole issue of placebo controls, and how do we handle it. And so his suggestion, I should add that probably half the time that of the committee meeting was spent talking about this exact issue. Because, you know, as you can imagine, once you start crossing people over, then you lose the randomization, things get very tricky. So his proposal, and it's not his original idea, I'm not remembering who first proposed it, but
Folman.
yeah, okay, thanks. Yeah, and yeah, and Anthony Fauci proposed this kind of. It was in the New York Times, so it's gotten some attention. The idea is the blinded crossover study, where you go through the first vaccination, and then at some point you just say, we're going to revaccinate people. So everyone who was on placebo has the option to take the active vaccine, and everyone who was on the active vaccine has the option to take the placebo vaccine. But nobody knows, nobody is unblinded at this point. So only somebody in the coordinating center knows who's getting what. So the advantage of this is that it does at least maintain the randomization to some extent. It maintains the blinding, which is, I think, maybe the primary advantage, which is when you're into subjective assessments of adverse events in particular, maintaining the blind has some advantages. It does not really maintain the randomization in terms of being able to make comparative effectiveness or comparative safety studies, other than the kind of pre-post. If you look at people who were on placebo, you know, what happened to them, what were their adverse event rates before and after the real vaccination. So I just want to toss that out as an area of a lot of discussion and a lot of concern. So, you know, these studies, when we were first discussing them, had 2 years of planned follow-up. And a bunch of us looked at that and said, you're not going to run a 2-year placebo group in the middle of a pandemic. So we're trying to find creative ways to address these questions. So one of which is this, and this is where I'll expand on what Courtney was suggesting, which is, and she really laid out the options pretty well in terms of linkage to the participants in the trial, linkage to these readiness cohorts. One of the issues that comes up, especially once a vaccine is out there and you're getting into actual clinical practice, one of the main issue that has come up is, what do you do for a comparison group? The obvious one is to take people who are not vaccinated, but as we've heard, there's a fundamental issue with people who decline vaccination being very different potentially in terms of risk factors and behaviorally from people who take the vaccine. There's an interesting website, which I'm not going to remember off the top of my head, but it's got a graphic that looks in the US by state at Republican versus Democratic Party registration, and looking at things like social distancing and mask use, and infection rates. And political party turns out to be a pretty strong predictor of what goes on. And so there's a bunch of stuff, a bunch of behavioral stuff that does not get captured in any of the existing databases. So if you want to use available data, then you need to come up with ideas for how you might really address confounding. And I'll be interested to hear Jamie's thoughts on that in particular. Another point, and I'll be quick with this one, is just basic epidemiology is how do you count person time? And I know there are solutions to this that have been used in the past, but one is for multiple dose vaccines. How do you attribute, if there's an adverse event, which dose does that get attributed to? But the compelling example for me, this is discussions with Patrick Ryan and my group, is anaphylaxis, which has been in the news. How do you count, or do you count person time when you're looking at anaphylaxis? And it's one thing in the context of a trial, but if you're trying to come up with background rates from the population, it takes a lot of thought. It's a tricky problem because it's some specific encounter that would generally lead to an anaphylactic reaction. Start of a new drug, a vaccine, there's something that triggers it. So you can't just start counting up all person time. You have to have some way of defining kind of an index date. And when you do that, things can look very different from if you just count all of the person time. And I'll stop there. I think I was under my 10 minutes. All right, thank you.
Thank you, Jesse. Those are all very thoughtful ideas to continue the discussion with. And as next up would be Paul Copeland, also from Johnson & Johnson. Paul, do you have anything you'd like to add to the discussion? I think you're on mute. Could it be you're on mute?
Yeah, thanks. Yeah, Bets, I do have a few comments that we just wanted to go through. Firstly, about how we use modeling at Johnson & Johnson to select clinical trial sites. And secondly, a little bit on endpoints, if I have time. Just by way of introduction, I lead the epidemiology and real-world data sciences team for medical devices at Johnson & Johnson, reporting to Jesse. But I've worked on the development or post-approval studies for, I think, 14 or 15 vaccines over the last 25 years. So we use modeling extensively for site selection in the Johnson & Johnson efficacy trials, in part because one of what Natalie referred to about the Ebola vaccine study that got caught in this end of the epidemic during the course of the trial was actually the Johnson & Johnson vaccine, which, as Natalie said, it did get approved, but it took a long time to get approved because we didn't really have enough, we weren't adequately powered to see for the end of the trial. So our team was very concerned that we would be caught at the end of the COVID epidemic and we wouldn't have enough cases. So we did extensive work on using modeling to help predict where to locate sites and countries. In addition to the work that Courtney mentioned around trying to get readiness cohorts that could feed in people at high risk within the sites, and also ethnically and racially diverse populations. But the way we use modeling was first to select sites, and secondly for the incidence calculations for the sample size estimation. And if the incidence that we used to select countries, that was our first step, to select countries. And to select countries, we were assuming that the US would have a big epidemic in the spring and early summer, But by the fall and the winter in the Northern Hemisphere, that epidemic would largely be controlled in the US. That turned out to be an incorrect assumption. And so we were looking at more geographic diversity for countries that would be less able to control the epidemic. Turns out currently, as the Johnson & Johnson trial is still ongoing, the US is the highest incidence of all the countries that we included. Anyway, but then the second thing was within the US and within countries to select locations. So for example, within Brazil, as Natalie showed, which provinces, or within the United States, which cities or counties. So the approach that we used was, I think, a three-pronged approach. Firstly, we held a think tank to review several different models with a bunch of modeling experts to get input on those models. Once we held the think tank, then we worked with those modeling teams to really review and understand their models and pressure test their models to make sure that they were there weren't deficiencies in the model. And then thirdly, we worked with local infectious disease experts to kind of get some face validity on the models, particularly for local regional issues. And that's how we connected with Claudio and to help us with validating some of the model output for Brazil. So the think tank, we included people like people from London School of Hygiene and Tropical Medicine, John Edmonds and Mark Jitt, Imperial College, Azra Ghani, Chris Murray from the University of Washington through the Institute of Health Metrics and Evaluation, Mark Lipsitch from Harvard School of Public Health, the Gates Foundation, and then also Dimitri Batsimas from MIT. And what we found was there were three models that we thought were the best models to use. MIT was one of them with Dimitri Batsimas, and then IHME model with Chris Murray, and another model that was developed by the Gates Foundation that a group called the COVISM model that a group at the Burnett Institute in Melbourne would run for us. And we also triaged with the CDC ensemble model to kind of validate what we're seeing from these models. And also with the Operation Warp Speed, also did some of their modeling and triaged with them to kind of get some checks and balances on our modeling. What we found in one of the challenges was the validation of the models, because the IHME model used deaths the goal of the model was to predict the incidence of infection in the population, because that's what we would be largely measuring in the clinical trial. I'll get into that in a moment. But so what data was available to measure, to approximate infection was the test positives. So those people who were getting tested positive. So those surveillance data were available. The number of deaths, which is also captured by surveillance, and then seroprevalence surveys in population seroprevalence surveys. But none of those actually capture the incidence in the population. So the difficulty in validating these models as we were approaching the clinical trial was what validation metric do we use to test whether the prediction models are in fact accurate? So what we did was we landed up testing the models against all three of the measurable outcomes that we had: the test positives from the surveillance test, those people who had gone in and gotten tested positive, the deaths, and also against the seroprevalence survey. So we looked at where we would get seroprevalence surveys, looked at the correlation between the model predictions, where we looked at model predictions several weeks prior to the actual seroprevalence surveys, and looked at how the model would have predicted the cumulative incidence from the model correlated with the seroprevalence data. The other challenge we faced was that of evolving model fits. So as the model started to introduce newly available data, they evolved quite substantially. So some of the new data that became available was social distancing from mobile phone surveillance data, and also mask wearing surveys from Facebook. And also some of the death data improved. So as some of the models added these data, there was the models had some issues with convergence, and we would get these very varying estimates over time. So what we found in retrospect, when looking at how valuable the models were in predicting where the incidence was during the trial, which we currently now have, the actual incidence from what's going on in the trial, that the models were good for selecting sites, but not so good at estimating incidence. So they were good for like where to go, but not so much for the sample size calculation. They were also good at estimating a general trend, but not very good at the timing of the trend. So oftentimes they predicted outbreaks, the models predicted outbreaks later than they actually, sorry, before they actually occurred. So what we actually had to do was we estimated a sample size of 60,000 people, and we ratcheted that down to 43,000 based on the actual incidence as the trial was ongoing. The other issue we faced was that the endpoint for the
Paul, I think we're getting about to your time, so.
Okay, I'm at 8 minutes and 26 seconds, but I'll stop there. That's great. Thanks.
No, that's fine. It's just that the 10-minute was sort of somebody made that up. But we have 3 more discussants. So, but those are really, I'm sorry to interrupt, but those are really interesting points. And Dennis wants to ask a question, but I think we're going to hold the questions till the end. But that was very comprehensive. So next is Jamie Robbins. Jamie, did you have any comments you want to make? Jamie, are you there?
Yeah, I'm here. Not really, because I'm not expert on this, but I have some questions. In the trials, for example, that have now been licensed, is there any way, and what are people doing about trying to learn what the actual infection rates were, like rather than clinical, infections, just, because people are worried about transmission and so on. Are there ongoing ways, to look at that, in terms of whatever they might have?
That was actually the point I was going to make in my 2 minutes of discussion.
Oh, okay.
That was going to be one of the challenges I'll bring up in my discussion, because the answer is no, in basically no.
And secondly, in the, again, with the ones that have been licensed, what's actually happened with the placebo? What were the actual decisions about what to do about people who are on placebo? I've heard the discussion, but I don't know how it turned out. Have there been decisions made about offering crossover to people on placebo? And I guess finally, I wasn't quite clear about the real world data issues, linking people to health systems. Does anybody have any sense of what that captured and what it didn't capture in terms of even events? Let's say non-hospitalized events or something. I mean, you're protected by randomization, but what do you actually know from those kind of, must be very disparate kinds of systems, if that was really the way clinical outcomes were assessed through various local healthcare practices? That's what I have to say.
Well, those are all good questions, and we're going to continue, and then we'll open it up for a regular discussion. I have, just because you brought it up, I'm the next on the list of discussants. And I'm only going to make one point, is that these trials really are not, the phase 3 trials are not set up to look at transmission. And that's a real issue. That's a real challenge. And there were some attempts to design a study that was going to be in college students in the US, but NIAID decided not to fund it. It was going to maybe go to the field in January to look at groups of people and look at contacts, because you really need to have transmission units to look at transmission. So these phase 3 trials are not looking at that. They are looking to some extent at infection, because they're doing some measures on serology to see if people got infected, but it is another challenge that's not really well understood, because the outcome, as you all know, is based on disease, on clinical disease. And it's important both for policy and for modeling to understand the effect on transmission and also the effect of the vaccines on infection, to be able to put these into models to look at really what the population level effects are. So that was the one point I was going to make, and we can discuss that if anybody wants to. And then the final discussant is Claudio. So Claudio, do you have any comments or questions you want to raise before we open up for general discussion?
Yes, Bets, thanks. First, thank you very much for Natalie and Courtney for the wonderful talks that they gave. And so my comments will touch indirectly in what they said, also Jess mentioned, and what Jamie is asking. So if we go to the very definition of vaccine efficacy, I guess that there is a lot of misunderstanding about its meaning. And the situation is similar to the situation we had with R naught, the basic reproductive number. So suddenly we are all talking about vaccine efficacy in the same way that we were all talking about the basic reproductive number. And we all want to maximize this efficacy in the same way that we would like to have R below 1. So if I go to my news web page, I can see that every day that there is a new vaccine, and with the trial reporting numbers such as 90% efficacy, and another one reporting 70% efficacy. And so those numbers taking at the face value would lead me to think that the 9% vaccine, efficacy vaccine, would be preferable to the 7% efficacy one. And this might not be exactly true for several reasons. The most obvious one is that the way that people are counting the numerator is a different, they are counting the people that enter in the numerators of those statistics in a different way. One is repeatedly checking to see if people get infected, and the other one checks only if there is some symptom signaling that they should be tested. So, but this is the easy one. The one that is more tricky is the one that people touched previously, and it has to do with the interpretation that we have of vaccine efficacy. So the interpretation that I see people making is that if I get infected, which means that if I get challenged by the virus or the pathogen, then this efficacy number has to do with the decrease in my risk of becoming ill. And so there is this conditioning going on that we cannot recover from the figures that are actually being reported. So there is this mismatch between our interpretation and what is actually being estimated. So what is being estimated is somehow an integration of this life history of challenges that people in those trials are receiving. So you have people that are not challenged at all. You have people that are challenged just once. You have people that are challenged more than once. And as Jesse mentioned, it's not just a matter of being challenged, but also the pattern in which they are being challenged. So if I have two participants that got challenged twice, if the challenge times are very close to each other, it's completely different than, or might be different, if they were challenged far apart from each challenge. So the history, the actual history of this challenging pattern has to do with the side effects, and it has to do with this estimate of efficacy. So I guess that the question here is, when this integration over those life history of challenging can be reconstructed from the kind of information that is being generated by those trials. And as we saw, I mean, this is not something that we can do easily unless we start collecting more detailed information about how people are getting challenged. So this idea is related to what Beth and I did a long time ago when we were facing this kind of interpretation, that we came up with the concept of a biological efficacy, which is this probability of getting sick given this history of challenge. At the time, we were thinking on just one challenge, but now we see that it's not a per challenge measure, but it has to be a per life history of challenging pattern. And so this is one challenge that we have in order to interpret this, those efficacy measures. The solution for that I see is related to the readiness cohorts that Courtney mentioned, but with the addition of a more detailed prospection of how people are being challenging. So we have to reconstruct this transmission network. And one analogy that we could make, or one idea that we could invoke from this discussion that is going on about the use of volunteers to artificially, that could be artificially challenged, so is to, instead of using these people, instead of designing the study, the trial in such a way that they would be artificially challenged, which I find very difficult to do, is to have their consent in such a way that we could reconstruct their infection life history, this challenging contact life history, even if this could somehow imply in some sort of privacy consent. So their volunteering would be in allowing us, in allowing the studies to assess this information about transmission contacts. So this is one sort of comments. And the other one is related to the evolutionary aspects of these vaccine studies and the information that we have to recover in order to understand what's going to happen with the use of those vaccines. So one first idea is that Natalie mentioned Zika and chikungunya, which are vector-borne diseases. And we will soon have two sorts of vaccinations going on at the same time. One is vaccination of humans, and the second one is vaccination of vectors. So we have to think ahead of time how we are going to collect this information and evaluate those trials and assess the efficacy of each one of those trials, because they will take place simultaneously. And the second one, also related to this evolutionary aspects, is to remember that we can have four basic mechanisms of action of a vaccine. One is to block infection, the other one is to modify disease, the other one is to block transmission, and the fourth one related to vaccines that would deal with toxins released by the pathogens. And in 2001, Sylvain Gandon published a paper in Science in which he was discussing the evolutionary trajectories of each one of those mechanisms. And the vaccines that modify disease are the ones that are more prone to allow for the evolution of virulence in those contexts. So we need to keep this in mind, and we needed to incorporate this kind of discussion in our trials. So I guess I am under my 10 minutes, and this is what I had to say, and thank you very much.
Well, thank you, Claudio. And I'm not sure who organized the 10 minutes, but if we have two speakers at half an hour and
I was the one.
And five discussions at 10 minutes, then you don't get it in under an hour and a half. So, but thank you. Those are all very good remarks. So I want to say at this point, I mean, we organized this, and Claudio really organized it, but I was sort of behind him on this. So because almost everybody in our field is so incredibly busy. And so we organized a really big topic for a short period of time. Now, I'm able to stay on a bit longer if we want to have more discussion, and people should feel free to leave because everybody has a lot to do. But we'd like to open up the floor now for discussion, and we will follow up. I really want to thank the speakers, both Courtney and Natalie, for excellent talks and also the discussants. So we can open up the floor now to regular discussion. Like formally, we only have 5 minutes left, but we are going to continue this discussion by email, and if we need to, we will follow up by. So at this point, if anyone has something else they'd like to say, I'd like to open it up to the floor and for the last 5 minutes. And then if people want to stay on, I can stay on a bit longer myself. I don't think the Zoom turns off automatically, but we could turn off the recording at some point.
Could I just chip in? There's no problem on timing. This session can go on as long as people want, as long as anybody's prepared to be there. But you should give me a clue when you'd like the recording switched off. At the moment, it's still going. So
I think we can turn off the recording now.
Okay, so I'll do that.
Turn it off.
But in terms of timing for the
right, I can stay on for another 15 minutes or so, and so we can open up the floor. But partly the short time